Okay, so this is the lesson on analgesics, antibacterials, and antivirals. This is 11.2, but in the textbook it corresponds to D2, D3, and D5. All right, so when we talk about analgesics, these are just drugs that relieve pain. And there are two classes, mild and strong. So we're going to talk about mild first and then strong. Now, mild analgesics include aspirin, ibuprofen, and paracetamol, which you commonly know as Tylenol. But you are only required to know about aspirin for the IB exam. Now, how do these work? These intercept pain at the source. So they're going to interfere with production of substances that cause pain, swelling, or fever. Aspirin specifically suppresses the production of prostaglandins. Now, we also know from our discussion in 11.1 that aspirin acts as an anticoagulant, and so it can be used as a preventative medication for heart attack and stroke. We also know that a possible side effect is stomach bleeding. Now, the risk of stomach bleeding is increased when aspirin is consumed with alcohol. So this is an example of a drug interaction or a synergistic effect. So synergy is like working together, right? So synergistic effects occur when two drugs interact together and create some kind of new or enhanced effect. So sometimes we're also just really intensifying their effects. And one interesting thing about synergistic effects is that it's not always two medications. So for example, there are some medications that you aren't supposed to drink grapefruit juice when you're taking them because it enhances the effect of the medication to an unsafe degree. Or it could decrease the effect of the medication potentially. All right, so when we look at the synthesis of aspirin, it's first, the starting material is salicylic acid, which comes from willow bark. And it's reacted with ethanoic, also known as acetic anhydride. And we're basically in acidic conditions. It's basically an esterification reaction on the hydroxyl group here. And we produce ethanoic acid as a byproduct. So this product on the right is aspirin. So we've just replaced the hydroxyl group with an ester. Now, after we make the aspirin, we have to purify it, obviously, before it can be used. So it goes through a few steps. Next, we have crystallization. So basically, you heat the mixture and dilute it with water, and then as it cools, it produces crystals. However, that's fairly impure, so then they use hot ethanol to recrystallize it. Once we think we have a relatively pure product, we would characterize it. So we could use IR spectroscopy to look at the functional groups that were present in the molecule. And we can also do a melting point determination. So for example, let's say I have a substance that has a melting point of 125 degrees Celsius, and I want to know if it's pure. If I do a melting point determination and I find that it's melting at 120 degrees Celsius, well, that indicates to me that I have some impurities there. Okay, the closer I am to the known melting point, the more pure the sample likely is. Now, aspirin has fairly low solubility in water, which limits its bioavailability. But we can increase this by converting it into an ionic salt. And it's not just aspirin we do this with. This is actually a pretty common technique to increase bioavailability. So this is our aspirin molecule, and we're targeting the carboxyl group here. So we're going to basically neutralize that carboxyl group with sodium hydroxide, and we're going to remove the acidic hydrogen, and now we're going to have an ionic salt. So we have the negatively charged group here that's forms a compound with a positive sodium, and we produce water as a byproduct. Now, drugs with amine groups can react with HCl, and so here we deprotonated that with sodium hydroxide. We removed a hydrogen. When we add HCl, we protonate the amine group, so we add a proton, creating a positively charged ion that interacts with chloride. And so an example of a drug where this is done is Prozac, but if you look in your medicine cabinet at home and actually read the drug labels, you'll probably find at least something that says name of the drug, HCl, because this is a pretty common technique. So let's talk about strong analgesics. So these are available only with a prescription because they include things like morphine and codeine. Diamorphine is commonly known as heroin, and that's a semi-synthetic drug that's derived from morphine. Now, these kinds of medications, so morphine and codeine, are used to relieve severe pain. So this is caused by a serious injury, surgery, heart attack, or chronic disease. And these drugs work by binding to opioid receptors in the brain and actually blocking the pain signals. So the mild ones worked at the site of the pain, but these actually block the signals in the brain. So morphine and codeine are called opiates, and they're derived from the opium poppy. And all of these drugs contain the same active area, and we can synthesize both codeine and diamorphine from morphine. So looking at the synthesis of codeine, morphine, and all this ring structure, this is the active area that's going to be common to all of these drugs. And also, all of these structures are in the data booklet, so just know that they're there. You're not by any means expected to memorize these. And so we're targeting this hydroxyl group here. So we would react morphine with methyl iodide, and we would create codeine, which instead of the hydroxyl group, we now have an ether and hydrogen iodide as a byproduct. Now, when we synthesize diamorphine, we're targeting both hydroxyl groups actually, and we'd be reacting it with acetic anhydride again. So this is the same reagent that we used to synthesize aspirin from salicylic acid. And we're converting both of those hydroxyl groups into esters here. So in both cases, what we've done is we've taken these relatively polar hydroxyl groups and made them less polar. Now, the advantage of that is that it's much easier for these to cross the blood-brain barrier. Diamorphine in particular is now much less polar than it was, and so it crosses the nonpolar blood-brain barrier really, really readily. And then the side product here is 2 molecules of ethanoic acid, or 2 moles. All right, so when we compare these structures, the physiological activity of these drugs really depends on their activity to cross, or their ability to cross the blood-brain barrier, because they have to reach the opioid receptors in the brain. So when we look at codeine versus morphine, codeine crosses the blood-brain barrier really easily, but that ether group that we added in its synthesis prevents the bond to the opioid receptor. So codeine works by slowly metabolizing into morphine. And because it metabolizes fairly slowly, it's about 10 times less potent than morphine. It has low activity, a wide therapeutic window, and a limited potential for abuse, so it's a pretty attractive choice for treatment. When we compare heroin and morphine, we made it a lot less polar, so it's soluble in lipids and it really easily crosses the blood-brain barrier. The difference between heroin and codeine is that heroin metabolizes very quickly into morphine, so the effects are much stronger than they would be with morphine or with codeine, which is being metabolized really slowly. The net effect of this is that heroin is 5 times more potent than morphine because it crosses the blood-brain barrier so quickly. And this also means that it has more severe side effects and withdrawal symptoms. So heroin is not a therapeutic drug. It's a really dangerous substance because it's so potent. And it's responsible for nearly 50% of all drug-related deaths worldwide. Now, when we talk about the side effects, opiates do have therapeutic effects. Morphine and codeine do have legitimate uses in medicine. They're strong painkillers, they provide euphoric effects and relief from distress. So if you were in a very severe accident, morphine would be an attractive choice to treat pain because it would have a strong pain relieving effect but also calm the distress. But the side effects of opiates can be really severe, build tolerance very quickly, they're very addictive, and they can depress the central nervous system. So for more mild pain, opiates are not going to be the drug of choice because there are some serious risks that come along with using them. And again, a very high potential for misuse. All right, so let's talk about antibacterial drugs. So antibiotics are used to treat infections caused by bacteria, and that is generally widely understood in the science community, but a lot of people want antibiotics for everything, which is part of the problem of antibiotic resistance that we'll talk about in a minute. So let's talk about how we came to use antibiotics. So first, we had to discover penicillin. So Alexander Fleming basically left an open petri dish and went away. But this isn't a good example of serendipity in science because when he came back, he noticed there was a mold growing on the dish that inhibited the growth of bacteria, and that mold was penicillin. So he published his work, but then other scientists actually took his work further. They isolated it and purified it and then used it to treat sepsis and learned how to make it in bulk. So Fleming discovered it, but then other scientists commercialized it, I guess you would say, and figured out how to use it for a practical purpose. So penicillin contains a 4-membered beta-lactam ring. So you can see that in the structure up here. And because it's a ring, the sp3 hybridized nitrogen, because there is a lone pair on that nitrogen, and the sp2 hybridized carbon here, the one that has the carbonyl group, can't achieve their normal bond angle. So for the sp3 nitrogen, it would probably be 107.5 since there's a lone pair. For the sp2 carbon, it would be 120. But again, it's a square ring, so they're going to be sort of compressed to 90 degrees. So what this causes is what's called ring strain. So they're being compressed to sort of an unnatural bond angle, so it's very reactive, and it's very easy to open that ring. So this makes the amide group here very reactive. And the group that contains the ring does have structural similarities to cysteine and valine. So when the ring opens, these areas bond to the enzyme that makes the bacterial cell walls, that enzyme is called transpeptidase, and disrupts their construction. Now, penicillinase is an enzyme that's responsible for some antibiotic resistance because it breaks that beta-lactam ring before it can reach the target molecule. So what we've done to prevent this is modified the side chain, so that R group there, and that prevents penicillinase from breaking the ring until the correct time. Now, antibiotic resistance is a huge, huge problem, and there's a whole host of causes for this, but one is that antibiotics were initially used to cure really minor illnesses. You have to imagine when we've never had a cure for things like sepsis or even things like strep throat, all of a sudden when antibiotics appeared, people were really, really enthusiastic about them, so used them for just everything. And as a result, certain bacteria developed resistance and then were able to multiply. So chemists modify the side chain, and they've modified it many, many times in many, many ways, but bacteria multiply and mutate very quickly, so we're sort of in a continual race to find new antibiotics. This is particularly a problem in the treatment of certain diseases, tuberculosis being a notable example. And so people with these infections often have to take multiple antibiotics in very precise regimens to halt the risk of further resistant strains. And you hear about this on the news sometimes, like superbugs that are resistant to everything, and that is a real legitimate threat. This problem is also compounded by the fact that we use antibiotics in livestock feed. So we eat antibiotics in our meat and dairy, which again promotes resistance. They're widely used in a lot of things. This isn't as common anymore, but for a long time, a lot of hand soaps had very low doses of antibiotics in them, which again just really promotes resistance. It's not necessary to have antibiotics in your hand soap. All right, so lastly, let's talk about antiviral drugs. Now first, we have to talk about why viruses are harder to target with drugs than bacteria. So viruses are basically nucleoproteins. So they have RNA or DNA that's surrounded by a capsid, which is a protein coat. So unlike bacteria, viruses don't have metabolism or cellular structure. They're not cells. So they can only reproduce inside host cells. And a really simplified version of how this works is that the genetic material of the virus sort of takes over the machinery of a cell to form new viruses, which then go on to infect healthy cells. So the body's natural defense mechanism is usually strong enough to fight viruses. So you've had a cold before and you know that you're sort of miserable for a few days and then you just get over it. Drugs are mostly used to alleviate symptoms. So over-the-counter cold medications don't actually fight the virus directly, they just reduce the symptoms. But there are now antiviral drugs that have been developed to fight specific viruses. So you hear about this specifically with fighting the flu. And these drugs can work in a few different ways. So they can actually alter the cell's genetic material so the virus can't use it to multiply, or they can prevent the viruses from multiplying by blocking enzyme activity within a host cell. So we're going to talk about a couple medications. Tamiflu and Relenza are the common names, and you've probably certainly heard of Tamiflu because that's a pretty common drug. And these prevent the release of virus copies from the cell by inhibiting the viral enzyme. So basically, the copies just don't go on to infect other healthy cells. So the one on the left is Tamiflu and the one on the right is Relenza, and these are given in the data booklet, but again, they're going to have their proper names, which I will try to say and butcher, presumably. Oseltamivir and Zanamivir, I'm guessing there, but that sounds reasonable. Now, when we look at their structural similarities, both of these contain a 6-membered ring that has 3 chiral carbons, and I'll identify those for you in class. It's a little hard to do via video lesson, but you could also presumably identify them yourself. Now, when we talk about structural differences, there's many different functional groups in the side chains. So a really common question that IB might like to ask is, what functional groups are present in zanamivir and not in oseltamivir, right? And then you just have to look at the structures and find them. Or they might even say which peaks would be shown in an IR spectrum of one versus the other. So not necessarily super hard questions, but you do need to review your functional groups. So for example, here we have a carboxyl group that we don't have in the other molecule. We have numerous hydroxyl groups here. We have amino groups in both molecules, and then we have esters in both molecules as well. Now, one of the esters in oseltamivir, this one right here, makes it inactive in its original form. So it's the only ester. There's no ester in this molecule. There's just a carbonyl. This is the only one with an ester, but it makes it inactive in its original form, and then it's hydrolyzed in the body to create an active metabolite. All right, so why is it difficult to target viruses? So viruses, like bacteria, rapidly mutate. An example of a virus that we've had enormous trouble targeting is HIV, which causes AIDS. So HIV is a retrovirus, which means that it contains RNA. It invades healthy white blood cells and makes DNA from that template using reverse transcriptase. One drug that's been somewhat successful in combating this is zidovudine, which binds with the reverse transcriptase so it can't make the HIV DNA. And this is reverse transcriptase is primarily just used by retroviruses, so it doesn't hurt the normal cells. There are, however, other challenges besides drug development in fighting HIV and AIDS. So for one, in many places in the world, and even in places that have access to healthcare, not everyone has it, limited healthcare is a huge issue. The drugs that do fight HIV and AIDS can be very expensive, and depending on how healthcare is run in that country, access to them can be challenging, or access to preventative medications could be a challenge. Poor education and awareness. So there's a lot of people that don't realize that they have it, and so they can pass it to other people without being aware. And in some places there's poor education about how HIV is spread, or various ways of dealing with it, and so education and awareness can be a real challenge. And finally, sociocultural issues. So this could be just a wide array of things. Some cultures sort of reject the use of certain preventative treatments. Illegal drug use can be an issue that leads to the spread of HIV, things like prostitution, and there's a huge stigma around HIV. And so in places where there's a huge stigma, people may be reluctant to seek treatment because they don't want to be identified as being infected with HIV. So there's also some cultural issues around treating HIV that can be really, really challenging. Okay, so do the homework, read the textbook. That is the #1 best advice you can get for this entire unit, and ask me questions in class.